Hi guys, welcome to Easy Bio. Today we are going to talk about different syllabus, which is national syllabus in Egypt or Egyptian national syllabus. The first lesson in Senior 1 is talking about the chemical basis of life. So we need to talk about the chemical structure of living organisms' body. What are these chemical structures and what are their function? What are they formed of? Before starting this part, we need to know what is the structure sequence of living organisms. As we know, if we take the human being as an example, so the human being considered as living organism. So we have the whole organism. This organism formed of group of systems. So your body as a human being formed of digestive system, circulatory system, formed of nervous system, and so on. So the whole organism formed of different systems, and each system of these, if we take the example as digestive system, each system is formed of group of organs. So what are the organs of digestive system? For example, we have mouth, we have esophagus, we have stomach, we have intestine, we have anus. So these are the organs of digestive system. If we take each organ and see, so each organ formed of group of tissues. The stomach, for example, formed of different layers. Each layer is considered as a tissue. So these tissues, like epithelial tissue, we have epithelial tissue which lining the stomach. So we have group of tissues. If we take each tissue, it will be formed of group of cells. We have the epithelial tissues which is formed of epithelial cells. So each cell of these formed of organelle. We have the animal cell formed of different organelles. They formed of nucleus, cell membrane, cytoplasm, mitochondria. So these are called the organelles. And each organelle of these formed of group of molecules. What are these molecules? We have protein, lipid or fat, and we have carbohydrates. So these are the molecules which found in each cell. Each molecule of these, each chemical molecule of these formed of group of atoms. What are these atoms? Carbon, hydrogen, oxygen, nitrogen. So this is the sequence of the living organisms. So each one of us formed of these sequences. If we are trying to classify the molecules in living organisms, they will be organic molecule and inorganic molecule. What is the difference between organic and inorganic molecule? Firstly, organic molecules, we are calling them biological molecules. And by the way, they are large molecules. They contain carbon and hydrogen, these two atoms. So the carbon is found in organic molecules plus the hydrogen. They contain, sometimes we can add, these are the basic atoms which found in each organic molecule, but sometimes we have oxygen, sometimes we have nitrogen. What are the examples? Our examples: carbohydrates, proteins, fat, nucleic acid. So these are the examples of organic molecules. But inorganic molecules, these are the molecules, and please focus, which do not contain carbon atom. So without carbon atom, they will be inorganic molecule. What is the example? We have water, H2O. Do you have any carbon atoms in H2O? No. And for example, NaCl or sodium chloride. These are inorganic molecules which found in our body. If we take the biological macromolecules, and we said biological macromolecules means the organic molecules. So the other name of it, polymer. What do we mean by polymer? If we have small parts and you connect these small parts together to form a big part, so you are connecting the small ones to form a huge one, which is called polymer. So for example, if I have small parts like these, which are called monomers. Mono, by the way, related to the number of molecules, means only one. So these monomers combine together by special bonds like this ones to form large molecule. This one molecule, but large, because it formed of many monomers, which is called polymer, and the process is called polymerization. So we have biological macromolecules are called polymers because they are formed of small units which are called monomers combined together to form the polymer. What are the examples? We have protein. They formed of subunits, small units, monomers, which are called amino acids, combined together to form the protein. Lipids formed of fatty acids and glycerol, and carbohydrates formed of monosaccharides or glucose. So we have examples of the polymers. Biological molecules inside living organisms are classified according to their structure and function into four. These are the main part of our lesson. What are the macromolecules or the biological macromolecules in our body? We have protein, lipid, and carbohydrates, and finally nucleic acid. We'll start firstly with the carbohydrates. Carbohydrates, as we said, they are large polymers or macromolecules or biological macromolecules. They are formed of monomers. What are the monomers of carbohydrates? They are called monosaccharides. And as I told you, mono related to only one molecule of sugar. So the simplest units of the carbohydrates are monosaccharides or one sugar, formed of one sugar. Okay, what are the examples of carbohydrates? We have different examples. We have sugar or glucose, we have starch, we have glycogen, and so on. We have cellulose, many examples of carbohydrates. General formula of carbohydrates, and this is very important. Formula is CH2O all out of N. What is the N? N is the number of carbon atoms. So it means if we take example, this C around 6 carbon atoms, so the hydrogen will be 12, oxygen will be 6 atoms as well, like the carbon atoms. So what is the ratio of carbon, hydrogen, oxygen? Will be 1, 2 to 1. This is the ratio of the atoms in the carbohydrates. So do we have any examples of these carbohydrates? In the plant cell, we have an organelle which is called chloroplasts. Chloroplasts contain green pigment which is called chlorophyll. The chlorophyll used for trapping the sunlight during an important process which is called photosynthesis. During photosynthesis, the carbon dioxide and water combine together to form the glucose. This glucose used for the plant, but the excess of this glucose used or stored inside it in form of starch. So by the photosynthesis, the monosaccharide or the monomer is formed, which is called glucose, and stored in form of polymer, which is called starch. I hope it's clear now. If we are trying to classify the carbohydrates according to their molecular structure, so it will be simple sugar or complex sugar. So we have only two types of carbohydrates, but the simple sugar itself classified into monosaccharides or disaccharides. What are the examples of monosaccharides? We have glucose, fructose, galactose, and ribose. And we'll explain in details. Disaccharides like lactose, maltose, sucrose. Complex sugar, which are called polysaccharides, it means formed of long or high number of monomers or monosaccharides like starch, cellulose, glycogen. If we start with the simple sugar, before explaining what do we mean by simple sugar, we need to know their properties. What are the properties of the simple sugar? First, our example is the sugar which you use in your kitchen. So take a spoon of the sugar and try to taste this. Add this spoon in a cup of water. It will be dissolved or not? Yes, it will be dissolved. So it will be water soluble. Taste this cup of water after dissolving the sugar. It will be sweety. So it's sweet taste. And finally, they have low molecular weight. To never forget this properties of the simple sugar, was low. What do we mean by was low? They are water soluble, they are sweet in taste, and they have low molecular weight. So never forget was low. The simple sugar can be, as I told you before, monosaccharides or disaccharides. Monosaccharides, they consist of, by the way, I told you mono means only one sugar. But they are different from one to another in the number of carbon atoms. They have from 3 to 6 carbon atoms. 3 to 6 carbon atoms. And these carbon atoms connect together with water, with hydrogen and oxygen. As you see, this is our example. This is the glucose. We have all of these, by the way, these points are carbon atoms, as you see, and each carbon atoms attached or connect with hydrogen and oxygen to form the cyclic form of glucose. What are the examples of the monosaccharides? We have glucose. What is the name? Grape sugar. Please focus. We have glucose, G, grape sugar, grape, G and G. We have fructose, fruit sugar, F, to never forget this. Other examples, as I told you before, we have galactose, this found in the glands of milk, and the last one ribose. And by the way, ribose formed of 5 atoms which used for the ribonucleic acid. Okay, so this contained different than the others which formed of 5 carbon atoms. Disaccharides means di means 2. So mono plus monosaccharides together to form disaccharide. 1 plus 1 forming 2. Mono plus mono forming di. And this di connect together by glycosidic bond, which used for connecting the monosaccharides together. What are the examples? We have lactose called milk sugar, formed of glucose plus galactose. Second, maltose, malt sugar, formed of glucose plus glucose, so 2 glucose together. Last one, sucrose, cane sugar, formed of glucose plus fructose. If you focus, you will see lactose, maltose, sucrose, all of them are formed of one molecule of glucose, but the other sugar is different. This is the example of our example of sucrose. One molecule of glucose plus one molecule of fructose. They combine together with the glucosidic bond as you see. So we have the disaccharide. The next point, what is the function of the monosaccharides? The main function of monosaccharides is production of energy. In our cells, in the living organism cells, we have organelles, as we said before. These organelles used for different processes. For this part, we have a special organelle. This is the cell. We have the nucleus, and we have a special organelle which is called mitochondria. Inside the mitochondria, glucose is going to the mitochondria. Inside the mitochondria, by the help of oxygen, the glucose is broken down into carbon dioxide and water. But because we are breaking down the bonds in between the carbons, this is the glucose. And as I told you, these are the carbon atoms. Because of the action of oxygen, so the bonds between carbon atoms are broken down. So they leading to releasing of energy. So the process of production of energy happens inside the mitochondria by breaking down the glucose into carbon dioxide, water, and energy. The form of energy is called ATP. But before that, we need to balance this equation. We have 6 carbon atoms which found in glucose. So we need to release 6 carbon atoms and 6 atoms of oxygen to release this, but the process results in production of 36 ATPs. This is the form of energy which used for the whole processes. The energy which used for walking, the energy which used for sleeping, for eating, for doing your work, all of this for keeping us alive, all this need energy. And this energy, by breaking down the glucose inside mitochondria to get the energy, the form of this energy which is called ATP. Sometimes we are calling it oxidation of glucose, but the scientific name of this process is called respiration. And by the way, this respiration is completely different than breathing. Breathing, taking in and out the gases, but respiration means a process, chemical process inside the cell, inside each cell of your body, for breaking down the glucose to get the energy used for keeping you alive. The next point, complex sugar. Complex sugar, we said they are polymers. What do we mean by polymers? Again and again, they are formed of monomers which are combined together to form long and huge molecule called polysaccharide. What are the properties? If you remember the properties of mono or simple sugar, it will be completely the opposite. In simple sugar, we said they are soluble in water, but these insoluble. What about the taste? The other ones, the simple sugar, are sweet, but these are They don't have sweet taste. And the last one, what about the molecule, the molecular weight? Their molecular weight in the simple sugar, they have small or low molecular weight. Here they have high molecular weight. What about their molecular structure? If you focus in the diagram, you will see we have the glucose, which are called monomers, but they combine together by glycosidic bond, and many of them to form huge or large molecule, which is called polysaccharide. Our question in this one, please build up a table and show what are the difference between the simple sugar and the complex sugar. You can do it by yourself. Please do it, and if you do it by yourself, it means you understood. Practical work for this part. How can we detect the presence of simple sugar and the starch? Firstly, how can we detect the presence of simple sugar? By getting 4 test tubes, and in the first one add glucose solution, second one starch solution, third egg albumin protein, and the last one distilled water. Related to the distilled water, I need to understand this point, please. Distilled water, this is called control experiment. What do we mean by control experiment? This experiment which is used to compare between the positive and negative. For example, here I have the water. I'm sure that there is no sugar at all in the water, right? Because water is inorganic molecule. So if we add the indicator of the sugar in the water, it will give negative. For sure it will be negative because we don't have sugar. So we can take the water, which we are sure it's negative, and compare between it and the other test tubes. So that's why it's called control experiment. So we add these ones, and then we add equal amount or equal volume of the reagent. What is the reagent of the simple sugar? Is called Benedict reagent. By the way, its natural color is blue color. Okay, so for example, if I add in each test tube 2 cubic centimeter of Benedict solution, then take the test tubes and add them in water bath for few minutes and check the change of the color. Negative, it means there is no sugar. Positive, it means the sugar is present. What is the negative result? Blue color. The color of the reagent will never change. But positive, it means the color will be changed. So here we have, in case of sugar, in case of glucose, the color will be changed from blue into orange. By the way, to be clear for all of you, any change of color other than blue, it will be positive. Okay, but in starch is a complex. Yes, we said it's formed of monomers, but they are combined together, so it's complicated. It can be indicated by the Benedict. Egg albumin, it's protein. There is no sugar at all, and for sure the water, nothing. We don't have any carbon atoms at all, so we don't have any sugar. So the color in all of these will be blue, which is negative. So the sugar is not found in starch. There is no sugar in egg albumin. There is no sugar in distilled water. But here, yes, it's positive because the color changed. By the way, it's orange in color, but it changed other than blue because sometimes we have red, sometimes we have yellow. It's according to the concentration of glucose, concentration of the sugar itself. So again, what is the reagent for the simple sugar? The Benedict What is the color of the Benedict color? The Benedict reagent is blue. How is it changed, or what is the positive result? It changed into orange. What is the negative? Negative, it means there is no sugar, so the color will be as it is, blue. The last one, detection of starch. The reagent which used for detection the presence of starch is iodine solution. What is the natural color of iodine solution? Orange brown. So if there is a starch, the color will be changed other than orange brown. If there is no starch, the color will stay as it is. So we'll get the sample, the solution, which maybe contain the starch, add few drops of the iodine solution and check the color change. If the color is changed into blue black or dark blue, it means starch is found. No change in color, so the color stays orange brown, so this is negative, no starch. So if we take some samples like soybean, green apple, carrot, macaroni, wheat and bread, all of these they contain starch but with different ratios. So the change of color, the darkness of color will be different from one to another according to the ratio of the starch. But one important point which is needed in this point, these are solid. They are solid. So it's so hard to get a piece of the soybean or macaroni and just add few drops of starch. To be clear and to be easy handling with it, so you should grind them. Grind them, and sometimes you can dissolve them in the water to form food solution, to be easily treated with your reagent. These some tricky points which are needed for the practical work. Thanks for watching.